Okay, so restriction enzymes or restriction endonucleases, they're the same thing. But commonly, we just say restriction enzymes because it's a little bit easier to say. These are like a scissors. If you think of a DNA molecule as a string or as a thread, right? You can use these restriction enzymes to cut the DNA strand into multiple fragments. So they're really like a scissors and just cut, cut, cut. All right, now, so how or where do those restriction enzymes cut the DNA? This depends on which enzyme you are looking at, because these enzymes are extracted from a different bacteria, and they have their own recognition site. So what they do is they scan the DNA strand, and once they see their recognition site, for example, let's say I'm just going to make something up, right? This is not a real. Let's say we have a restriction enzyme restriction enzyme number 1. It can recognize AAGG. So anywhere on DNA, when you see AAGG, this enzyme is going to cut DNA right there. All right. So let's say it's going to cut between the AA and the GG. So if you have a DNA strand here that says AAGG, the enzyme is going to cut right here. So this is going to generate a fragment, correct? Delaney, did I lose you?
Yeah.
Okay, all right. And then let's say you have another AAGG right here. So the enzyme is going to cut right here. So this creates another 2 fragments, right? So this is fragment 2, and this is a fragment 3. And then we can do different things with these fragments, right? For example, the next experiment that you're doing, the gel electrophoresis, it uses electrical current to separate fragments with different sizes. And then we can use kind of that to compare DNA samples from different people, right? We can identify, let's say we collected some DNA sample from a crime scene, and then we can compare that DNA sample with DNA samples from different suspects, and we can see, which suspect's DNA matches the DNA fragment from the crime scene sample. So we have a lot of applications once we can break DNA to different fragments using the restriction enzymes. Okay, so that's one real world application using restriction enzymes. All right, now, so how do we solve this problem? So I give you some hint right here. For enzyme 1, it's going to, so the different colors represent the different nucleotides. Right? So enzyme 1 is going to recognize when you have 2 red nucleotides together.
Okay.
All right. So let's say we're using this strand to do enzyme 1. All right. Now it's going to scan through the DNA strand, right? And then whenever it sees 2 red together, it's going to cut between, right? This is, again, simplified. So you're going to cut between the 2 reds. So it's going to cut right here.
Mm-hmm.
And then you have another 2 reds. So it's going to cut right here. So this generates how many fragments?
2.
Huh? Is it 2?
No, 4.
Look again.
So it's going to cut to 1 fragment, but...
So this is a 1 fragment, correct?
Yeah.
Okay. Is this a fragment?
Yeah.
Is this a fragment?
Yeah.
So how many fragments?
So 3.
3, right? So if you cut at 2 places, that's going to generate 3 fragments. So the number, for enzyme 1 is 3. All right, now let's figure out the fragment size. So let's just start with the smallest one, because it says you have to put the size in order, right, from smallest to the largest. Okay. So how many nucleotides do you see in fragment 3? 1,
2, 3, 4.
4, very good. So 4. You usually should put a unit there. Right? So the unit is a base pair. We talked about this during the case study. Remember base pair BP? because you have another DNA strand, right, which is not shown here. And all the nucleotides are paired up, right? So this is 1 base pair, 2 base pairs, 3, 4 base pairs. So that's the unit we usually use to describe the size. So this is 4 base pair. What about the size of fragment 2? How many nucleotides?
9.
9. That's correct. 9. Very good. I'm skipping writing the BP. All right. Now the last one is very long, so I'm not going to count it here to save time. So you can count how many nucleotides are in that fragment later.
I think 22.
22? I think you're right. I think the first one is 4, 9, and 22. Very good. All right. Now let's look at enzyme 3. This is a little bit more complicated. So this enzyme can recognize a red and a blue. And it doesn't matter who comes first. As long as it's a red next to a blue, it will cut in between. So when you look at here, there's a red and blue right here, right? So the enzyme is going to cut right here. Okay. And next, here, blue and red, cut. And the next one, red, blue. It doesn't matter, right? As long as you have a red, blue, so cut. There's a blue, red, cut. Okay. Red and blue, these two, cut. Next one, blue, red, cut. There we go. So how many fragments do we have this time for enzyme 3?
6.
6. Let's count. 1, 2, right? This is a very short one. And then the third fragment is very short. It has only 1
I
nucleotide.
didn't include that one.
Yeah, so you didn't include that one. And then this one right here,
7.
4, 5. Yes, you're right. So that's 7 fragments. Oops. Okay, 7 fragments. All right, very good. Now, what's the size for the smallest fragment?
1.
1. Excellent. 1. What's the next one?
So
probably this one right
2.
here, right? 2. Yeah. And the next one?
It would be 5.
5, right? So fragment 1 has 5 nucleotides. 4 has 5 as well. And then 7 has 5 too, right? But you need to write out all the 5s because each one is for each fragment. So you're going to put 3 5s in here. Very good. And then we have fragment 6, right? That's how many, looks like 7.
Yeah, 7.
7. And how about 5?
10.
10? Yeah. So that's the answer for enzyme 3. All right. Now, for the last one, enzyme 2 and enzyme 3, it asks you to see note. So when you open the instruction page, it actually says enzyme 2 and then enzyme 3. So this means you are going to let enzyme 2 cut the DNA strand first. And then enzyme 3 will come in and then cut the fragments further.
So I have to put in the number of fragments, 2 numbers?
Yes, that's correct.
Okay.
Yep. That's it for experiment 2. And then next experiment, gel electrophoresis, like I said, all the answers can be found in the video.
Okay.
Yeah.
I think I got it now. Thank you so much.